I want to quickly praise Sally for letting me know that she's here. That's what I want you all to do. Follow the model that she has now set. If you show up a couple minutes late, no big deal. I know we all have different internet and connectivity problems at different times. Just let me know, send a message to the chat. That way I can correct my attendance. So thanks for doing that, Sally. People will hopefully follow your model if and when it happens to them. Today is January 19th, 2021, and we are continuing our discussion from Friday about enzymes. We've got the same objectives, the same essential question, and hopefully we'll be able to answer that essential question today. Again, it is, what does it mean for enzymes to have an optimal environment? Today is still Unit 1. Today is the 5th day of Unit 1, and I want to take some time now to let you guys know or remind you that we will be having an exam on Thursday. We're going to do a day of review tomorrow, but your review hopefully has continued since we began the unit, as you just kind of flip through your notes and check in on things, and you're doing your warm-ups and exit tickets, which provide a great opportunity for review. But we're going to dedicate tomorrow's lesson to reviewing. Then we'll take a unit exam Thursday. It will be our first unit exam, 100 formal points, big deal for your grade. And then Friday, we will debrief and give folks an opportunity to catch up. That being said, all of your assignments for Unit 1 will be due Friday at 11:59 p.m. All right, let's go ahead and jump into it. So we saw this slide at the end of our lesson on Friday after we had introduced enzymes. And I told you that in review, enzymes are three things. Number one, they are catalysts, which means that enzymes speed reactions up. They take reactions that would normally happen on their own, and they help those reactions to happen with less energy required and in less time. And we're going to see a graphic representation of that today. Number two, enzymes are specific, so they only work with unique substrates. The shape of the substrate is compatible with the shape of an enzyme's active site. So the active site is where the action happens. That's where the chemical reaction takes place. The reactant, otherwise known as the substrate, needs to be similarly shaped to the enzyme's active site in order for a reaction to take place. And that's what gives it the quality of specificity. That's what makes the enzyme, quote unquote, specific. And lastly, number three, enzymes are reusable. We're going to see an image that I put together. Hopefully it helps you guys today that will depict this reusability. But essentially, enzymes will go into a reaction. They will facilitate that reaction and catalyze it. They will speed it up. And then at the end, the enzyme should remain unchanged. It should remain unchanged. That way it can go back into the reaction and do the whole thing again. We're going to see an image that I think will help explain this to you all today. You may have heard me mention that enzymes serve as catalysts because they are able to lower the amount of energy that's required for that reaction to take place. There's a specific name for that energy. It is called activation energy. And activation energy is the amount of energy needed for a reaction to begin. So an enzyme works to speed up a reaction by lowering that activation energy. So again, hopefully you all are writing down what you're seeing in yellow, highlighted in yellow there. I love efficient and effective note takers, people who can return to their notes as we move through subsequent lessons. This is a cumulative course. Everything builds upon things that we will have prior and previously learned. So we need to have access to that information. Can't go in one ear and out of the other. Okay, so at the bottom, we can see a graph. Can someone tell me what information is on the y-axis of this graph?
Potential energy.
Excellent. Thank you, JT. So I guess you've got some graphing skills either from math or from prior science classes, and that's excellent. But we need to know that when we're looking at graphs, the y-axis is the vertical axis. It's the one that runs up and down. I think we actually talked about that. In this case, the information that's going to be graphed on your y-axis is potential energy. Potential energy is not something we talk really much about in biology, but it's a physical concept, and it also comes up a little bit in chemistry. In this case, potential energy is representing the activation energy. And then on the x-axis, which runs horizontally, we've got the quality time. Now, in this case, I'm not telling you exactly which units we're using to measure. I'm not telling you that it's seconds or minutes or anything like that. We just know that time is on the x-axis, which means as we move from left to right, we are increasing in time. We're kind of seeing how time moves forward. Obviously, we can't really move backwards in time realistically. So, all right. We're going to start off in a reaction, early on in the reaction, obviously with reactants, otherwise known as substrates. So that's what you start off with. Now we can see with this curve a depiction of a reaction that's taking place. We can see we start off with reactants, and as time goes on, the potential energy initially increases, and then it decreases pretty dramatically. And then we're going to end up with products at the end of the reaction. This reaction in red, the red curve, represents a reaction without enzymes. So this reaction is not benefiting from an enzyme. Do you all have a prediction about what might happen when we throw an enzyme into the mix? What would a curve or a graph with an enzyme look like? Any ideas?
Maybe we'll go up.
Okay, so thank you, Lisa. We will see, similarly, we will see another hill, just like we see a hill here. So the potential energy will initially go up, but the difference is it doesn't go up as much. So the reaction with an enzyme is represented by this green curve here. We can see that the hill is much shorter. So what happened here? Well, what happened is that the activation energy that's required without an enzyme is pretty high. But when we throw an enzyme into the mix, the activation energy is lowered. And that's what the second bullet point states. Enzymes speed up reactions by lowering activation energy. In this case, the green hill represents the use of an enzyme, and the red hill represents a reaction that takes place without the use of an enzyme. So the hill here with the green hill, the green curve, is shorter, and therefore it's easier and faster to climb. I like analogies, so we're going to zoom out a little bit. Some of you are athletes, I know, and all of you have been kids at some point. So perhaps at some point in your life, you've been at the bottom of a hill and you had to run up that hill, either for your athletic training or just because your brain was like, I really want to run up that hill. Or maybe you were sledding and you had to get to the top of the hill to come back down, whatever it was. But I assume we've all been at the bottom of a hill at some point. Going up that hill requires energy. Depending on how long it is, depending on how steep it is, it might require more or less energy, but getting up that hill is going to require some energy. Your muscles are going to be activated as you go up. Your breathing is going to increase. Your heart rate increases. Now imagine if somehow, some way you were able to look at that hill and be like, nope, too tall, too steep. I'm going to lower it. Now it doesn't require as much energy to get to the top. And you can do it faster. It doesn't require as much time either. Same thing with enzymes. Enzymes give reactions that opportunity to choose. Nope, that hill, that activation energy is too much. I need to lower it. That way I can do things faster and with less energy. Enzymes give reactions that choice. They say, we're going to lower the activation energy and make this reaction go faster. Okay, so this is a pretty simple graph, one that you might draw on your notebook or maybe come back to and add some colors to it. But the activation energy measures the energy not from the bottom here. Notice that these arrows don't go all the way to the bottom. It simply measures the amount of energy difference between the reactants. So the reactants are right here, right, on our y-axis. It measures the energy difference between the reactants and the top of the curve. So the potential energy of the reactants and the top of the curve. The difference between those two is called the activation energy. Okay. Okay, so simple question here. You guys can answer in the chat. Which of these reactions is using an enzyme? Is it the reaction represented by the dashed line or the reaction represented by the solid line?
Dash.
Good, thank you. Josue. It's the dashed line. We can also break this down as well. So again, notice that the activation energy is not measured from the x-axis. It's not measured from 0. It's measured from the reactants to the top of the curve. We see two curves here. Josue just told us that the dashed curve represents the use of an enzyme, aka a catalyst. And we can see that the hill is much shorter. You've got to climb the hill to start the reaction. If you don't get to the top of the hill, the reaction never takes place. Another analogy that I like to think of, maybe some of you have been on a roller coaster at Carowinds or Six Flags somewhere or some other amusement park. In order for you to get that thrill, at first it's going to take a long time for that roller cart to be pulled up to the top of the hill. And then once it gets to the top, basically gravity does its thing. And then you kind of, for the rest of the way, gravity is kind of carrying that roller coaster through the track. And you might go up a couple more hills and then down more and might go through some hoops. But to get that initial amount of energy to get through that whole track, you got to get to the top of this hill. Same thing in chemical reactions. Let's say that I've got this red dot, hopefully you all can see in your screen. Let's say it only has enough energy to get here on the graph. It's going to roll back down to the reactants. Okay, so my reactants can only move forward and become products if they can get to the top of this hill. Once you get to the top of this hill, then it's relatively easy. You've got plenty of energy to carry you forward to the products. But if you don't have enough energy to get to the top, unfortunately, you'll roll back down and the reactants will remain reactants. So you need this activation energy. It's the amount of energy that is necessary for the reaction to take place. Okay, now remember, reactions are going to happen regardless. You know, a reaction can still take place without an enzyme. It's just going to be a slow process. So these two people on the left, they'll eventually start a fire. If they keep creating that friction using that flint, they will eventually create a fire. But this person on the left is able to, on the right, is able to do it much faster. That represents the difference between using an enzyme and not using an enzyme. All right, these people will eventually start the fire, we hope. It's just going to take them a long time based on the speed that we see them moving here. This person over here is very easily starting this fire and doing it quickly. It's like an enzyme. All right, the other thing that I keep reminding you all about enzymes is that they are reusable. So as long as an enzyme's shape is not changed, it can be used in reaction after reaction. And that's why we call it reusable. We are going to talk today about what happens when an enzyme shape is changed and why that shape might be changed. But for the most part, enzymes can go through reaction after reaction after reaction after reaction without their shape being changed. So let's look at this. Here we've got an enzyme. We can see the enzyme's active site where the reaction will actually take place. Here's the substrate. We can see that that substrate fits pretty easily into the enzyme, fits perfectly into the enzyme. They've got compatible shapes. So what's going to happen? Well, this substrate is going to move into the enzyme's active site. Before we notice that, obviously, in a cellular environment, there won't just be one substrate. There will be a huge class of substrates. There will be a lot of them all over the place. And they're moving around, they're bouncing off each other. So this substrate is going to move into the enzyme's active site. The reaction will take place, and now we create products. In this case, this is a digestion reaction, which means that the original substrate gets broken down. So let's watch that again. We've got a heart-shaped substrate, which will move into the enzyme's active site. A reaction takes place, and we get two products. So we start off with a singular substrate. One more time, we start off with a singular substrate, and then we're going to break it down, creating two, or I guess maybe we would even say four products, however you want to look at it. But the good thing about it is the enzyme is not changed. We should notice that the enzyme is not changed. It looks exactly as it did for the reaction. So what we can do now is introduce that enzyme in another place in the cell. And it can undergo the same reaction again with a different substrate and create more products. Now that same enzyme is going to move from substrate to substrate, creating more and more products. As long as the shape is not changed, the enzyme is still going to be functional. That's what we mean by reusable. Okay. All right, so let's look at this. Based on this diagram, based on the diagram below, starch must be which of our four biological molecules? Is it A, a carbohydrate? Is it B, a lipid? C, a nucleic acid? Or D, a protein? Answers acceptable in the chat. There's Abina. Hi, Abina. Thanks for joining us. So is starch A, B, C, or D? I'm waiting for answers in the chat. There are really two hints, especially if you have your notes from last week. Thank you for that answer, Melissa. Oh, I'm sorry. Thank you for the answer, Abigail. Thank you for your answer as well, Melissa. Can we get some others? Thank you, Lisa. Anybody else? Thank you, Dijon. Okay, so a lot of people saying A. Thank you, Ashley. A lot of people saying A, and a lot of people are correct in saying A. But can I have someone unmute themselves and tell us, why is A the correct answer? Why is starch a carbohydrate? How do you know that?
Because it's a sugar.
Excellent. Thank you, Abigail. So starch is a sugar, and sugars are carbohydrates. Now there's another clue here that has to do with maltose that should tell us that it's a carbohydrate as well. What's the clue that has to do with maltose?
The last letter.
It says O's.
Yes, good. So I see Janelle said it in the group, and Lisa and JT came off of mute and said it. It ends in OSE. It is in OSE. Words that end in OSE typically are carbohydrates. They're sugars. Okay, so this enzyme here is called amylase. Amylase is going to meet up with starch, undergoes a chemical reaction, and it starts breaking the starch down into maltose. So maltose is a sugar, which must mean that starch is a sugar. I can't break a protein down into sugars. That's not how that works. Oh, and Ashley also says there's another clue as well. Excellent. The shape. What about the shape are you noticing, Ashley? You're 100% right, but what exactly do you notice here?
I forgot the name of the shape, but in the notes there's like a picture and then it says that's a carbohydrate.
Good. So she's looking back at her notes and she's got a graphic depiction or a pictorial depiction of this. It's called a hexagon. It's got 6 sides, and that represents glucose. So in this case, we can see these 6-sided figures representing sugars, monosaccharides. Excellent. No problem. Thanks for joining us, Santuan. I will get my attendance fixed. And you too, Abina. Thank you. All right. What was I saying? But I can't break a protein down into a sugar. I can't break a nucleic acid down into an amino acid. That's just not how it works. I can break a sugar down into another sugar. I can break a nucleic acid down into nucleotides. I can break proteins down into amino acids, but I can't really crisscross there. If I break them down enough, meaning I break them down all the way into their atoms, to carbon, to hydrogen, to oxygen, then I can start to reform new monomers. But a nucleotide is never going to combine with another nucleotide to make a protein. That's just not how that works. All right, so if I broke something down into a sugar, well, then that thing must have also been a sugar. Okay. So enzymes function really in one of 2 ways, at least for the purposes of this class. There are some more complicated enzymatic functions that might come up in an AP biology class or in a college level biology class, but for the purposes of this class, we just need to know about synthesis and digestion. We've talked about synthesis before. Synthesis means to take smaller things and use them to build up something larger. Oftentimes, we're building a polymer from its monomers. But we can also combine 2 polymers to make an even bigger polymer. Digestion is the opposite of that. We're breaking something down into smaller parts. Sometimes that means breaking a polymer down into its monomers. Sometimes that means breaking a polymer into a polymer and a monomer, or breaking a polymer down into a polymer and a polymer. It just depends on the size of things and what the enzyme's role is. We can see the process happening here. In synthesis, 2 separate and distinct things are combined to create one thing. In digestion, one thing is broken down into 2 separate and distinct things. There are other names for these processes as well. Sometimes you'll hear this referred to as a decomposition reaction. That's something that you'll learn about in chemistry. But to digest is to break something down, to synthesize is to build something up. Pretty simple. All right, so keep in mind what I was saying before that slide about how only certain things can come out of breaking down something else. So let's take a look at this. It says specific enzymes are used to act on specific compounds. Number one, protease breaks down proteins into what? What must a protein be broken down into? Excellent. Yeah, a lot of people jumping in. Janelle, thank you. Josue, thank you. Abigail, thank you. Proteins get broken down into amino acids because amino acids are the monomers of proteins. So protease breaks down proteins into amino acids. There's no need to memorize these enzymes. Don't worry about it. Amylase breaks down carbohydrates into what? Number two. What will a carbohydrate be broken down into? Look back at your notes from last week at that table that we talked about. Good, thank you, Abigail. Amylase breaks down carbs into monosaccharides. So if you're like, I have no idea where these answers are coming from, I don't think I've ever even seen these words before, well then you've got a couple things that you should consider doing. Number one, look back at the class recordings from last week where we broke this down. We talked about those 4 biological compounds. Thank you, JT. Another word for monosaccharides is indeed glucose. Or you can reach out to me for some individual help. We can spend some time one-on-one going over some of this stuff. Or you can get with another classmate and say, hey, can I see your notes from last week? I just want to copy them down in my own notebook. Or can we all get together and share a document for note-taking purposes? It's the 21st century. We're learning virtually. We are doing school virtually. So you guys at this point are pretty familiar with the virtual tools that are available to you. But if you are confused, there's nothing wrong with that. And if you had the wrong answer in your mind, there's nothing wrong with that either. But we don't need to remain in that state. Okay, so we don't want to remain in a state of confusion forever. We don't want to remain in a state of incorrectness forever. We can get these problems corrected now in the first unit and move on. Okay, number 3, deoxyribonuclease breaks down nucleic acids into what? Which should be the answer to number 3? Nucleotides, good. Excellent, excellent. Got a lot of people chipping in there. Nucleotides is correct. And then lastly, number 4, lipase breaks lipids down into what? This is kind of always the awkward one.
Glycine and 3 fatty acids.
Thank you, Lisa. Glycerol and 3 fatty acids. Good. All right, now, hopefully you all are noticing something. What do all of these enzymes end in? So we've got protease is an enzyme, amylase is an enzyme, deoxyribonuclease, and lipase are enzymes. What do they all end in? Thank you, Janelle. They all end in ASE. Protease, amylase, deoxyribonuclease, lipase. Okay, so that's another thing that you all should write down in your notes. Most enzymes, not all, but most enzymes end in ASE. That's the clue, right? We want to have these mental cues. When we see those 3 letters, ASE, we should be thinking, that's an enzyme, which means it's a protein, which means its monomer is an amino acid. And I know that enzymes either build things up or they break things down. And I know that enzymes are specific. And I know that enzymes are reusable. So once you pick up on that clue, you should have all this information start to flood your brain. And when you're taking a test, it's good to get that information on paper. That's what we call a brain dump. Instead of just keeping it all up here, dump it out onto the paper. All right. So we want to have these clues. When I see ASE, I'm thinking about an enzyme. When I see OSE, I'm thinking about a sugar. Okay, the other thing about enzymes is that they are very sensitive, which means that they only function in specific environments. So an analogy that I used with my last class, some of you have younger siblings, and you know that your younger sibling only falls asleep in really specific conditions. They need to be tucked in. They need to be tucked in by a specific person. They need to be wearing a specific set of pajamas. They need to have their pet laying at the foot of the bed, or they need to have a stuffed animal curled up in their arms, or they need to have a nightlight on. They need to have a glass of water on the desk. So all of these things need to come together in order for your younger sibling to fall asleep on time. Enzymes are really the same way. They need to have all these different conditions regulated in specific ways in order for them to work well. If those conditions are not set properly and an enzyme is placed in an unfavorable environment, that enzyme will become denatured. And we're going to define that word on the next slide. But if we place an enzyme in an environment that is unfavorable to it, less than ideal, that enzyme will become denatured. To denature is to change the shape of an enzyme's active site. Which prevents the enzyme from working correctly. So again, the active site is the portion of the enzyme where the substrate fits in and the reaction takes place and we create new products. The active site is where the action, the reaction happens. So if I change that shape, my substrate might not fit in anymore, or my reaction conditions aren't really attuned to that substrate. So changing the shape of the enzyme's active site is called denaturing that enzyme, and it prevents the enzyme from working. Denaturalization happens when the environment is not ideal. What do we mean by the environment? Well, we're talking really in this class about two things: pH and temperature. Those are the conditions that need to be regulated. Now, we've got thousands of enzymes that are constantly at work inside of our body cells. And all of them have different working conditions. Some of you have siblings who like to sleep with the nightlight on. Some of you have siblings who like to sleep with the nightlight off. Some of you have siblings who like to have their door open. Some of you have siblings who like to have their door closed when they're going to sleep. So everybody prefers different conditions. Same thing with enzymes. Some enzymes like a pH that is really, really acidic, and we'll talk about some of those. On the other hand, some enzymes prefer a pH that's very basic. Some enzymes prefer really, really hot temperatures. Some enzymes prefer cooler temperatures. But in any case, if I take an enzyme and I change one of those conditions, eventually, if I change it too much, that enzyme will become denatured. So if I've got a pH that prefers a really hot temperature and I cool it off, that enzyme is not going to work anymore. It will be so damaged that its shape will no longer be able to accommodate the substrate. Its active site is no longer going to work. So every temperature has that preferred set of conditions, just like every child has that preferred set of conditions to fall asleep. And when all of those conditions are set up just right, well, then you're going to get some good sleep. when all of the conditions are set up just right for an enzyme, that's called the optimum environment. And the enzyme is going to work really, really well. All right, so, I've got the night light, the door closed, my water next to me, my pet is curled up in my legs, it's the right temperature, my fan is on, my dehumidifier is going. I'm about to be knocked out for 11 hours. I'm going to get some good sleep. Same thing with enzymes. Place it in that right temperature with the right pH, the right amount of substrate, no competitors. That enzyme is about to do a lot of good work. And this all comes back to one of the most important central themes of our class, which is that shape determines function. Shape determines function. If we change the shape of an enzyme, meaning we change that enzyme's active site, well, then the function of the enzyme is going to be thrown off. Okay. So here's a look at the pH scale, and this is something you all will talk more about in your chemistry classes, but it is obviously still important in biology. So the pH scale runs from 0 to 14, and it directly measures how much hydrogen is dissolved in a solution. Indirectly, what that means is that it measures how acidic something is. So you all have heard the word acid. You know what an acid is, or you can think of some acids. Some things, however, are more acidic or less acidic than others. The more acidic something is, that tells you that there's more hydrogen dissolved in it. Now, acidic things typically are not good for the human body in high quantities. And some things that are really acidic, even in low quantities, can be very damaging, very harmful to our skin and to other tissues inside of our bodies. But on the other end of that, we have things that are called alkaline, otherwise known as basic. And basic things also aren't really good for the human body. They can irritate the skin or other tissues, or even in some cases burn through it. So we've got to be careful in any case, and we want to try to find ourselves here in the middle with neutral substances, like pure water. Pure water has a pH of 7. That doesn't mean that there's no hydrogen dissolved in it. That just means that there's an equal mix of hydrogen and something else called hydroxide. Things that are really basic have a lot more hydroxide than they do hydrogen. Things that are really acidic have a lot more hydrogen than they do hydroxide. So we want to have balance, equal amounts of both. That happens in pure water. But look at this scale and you see obviously a lot of things with which you are familiar. The acid that is in our stomach is very, very acidic. It's got a pH of 1, which means that if we were somehow to reach down our own esophaguses and touch our stomachs, you would probably burn your finger, because that acid there is really, really strong, and it's meant to break down foods, not meant to come into contact with your tissues. And this is why some people get stomach ulcers, because they make that acid even stronger and they start to wear away the protective layers of their stomach. And so it burns a hole in their stomach. It can be very dangerous. Then we've got things like lemons, which give us that sour taste. And they can also, if you put a lemon on a wound, it can burn a little bit because that acid is reacting. Same thing with vinegar, same thing with tomato sauce or tomato juice. Now we're starting to get into things that are more neutral. Now you might wonder why the colors are the way they are. We've got things called indicators, which can tell us how acidic something is. So, and we've got pH paper, which we can literally dip into a solution and it will come out and be a specific color. If it's red, that tells you that it's something that's really acidic. If it's orange, that's something that is acidic, but not quite as acidic. If it's green, that tells you that it's more neutral. Something that's a dark purple is really, really alkaline or very basic. All right, so that pH paper or indicator paper tells you how acidic something is, and the colors literally look like this. And then some people who might be suffering from heartburn, they might take some stomach tablets to kind of calm down their stomach and to balance out that acid. So the pH scale is something that is pretty cool. It's very useful, often used in science. pH is referred to often. Good to know. All right. So when we look at a graph of an enzyme, we're going to see a curve. And I'm going to show you one in a few seconds. At the peak of that curve, the enzyme is functioning at its best. This means that the enzyme is in its optimum environment. Now, the opposite of the peak is when it's at zero, when it actually hits the x-axis. That tells you that the enzyme is no longer functioning. That tells you that the enzyme has been denatured. So let's look at this curve. We don't know what the name of this enzyme is. That's not the point. We see the curve, it's moving up, and then it's going to move back down. So the y-axis in this case is telling us how well the enzyme is functioning. As we go up higher on the y-axis, that tells us that the enzyme is working better and better. As we move lower, that tells you that the enzyme's functioning is decreasing. It's not working as well. And then when you finally hit zero, when it hits the x-axis, the x-intercepts, that tells you that the enzyme is no longer working at all. It has zero function. Okay, so let's think about this. What's the optimum pH for this enzyme? Should be pretty easy to see. Thank you, Josue. Thank you, Abigail. The pH of 8 is the optimum pH here. At what pH or pHs is this enzyme going to be denatured? Be careful with this part. This part's a little less obvious. What are the denatured pHs? And while people are thinking zero is an example of a denatured pH, yeah, we can't even see zero on this graph, but if we could, that would tell us that the enzyme is not working there. Okay, so some people are saying 10.5. Good. At a pH of 10.5, the enzyme is no longer going to work. There's another place as well, though. 4.8, I think that's a great example, or a great estimation, great estimate, not estimation, great estimate. I think 4.75, but 4.8 is probably even better. So when we look at this, we want to be careful. Thank you, Sally, and thank you, Abigail. We want to be careful here because we're not just looking for that specific point when the curve hits zero or when the curve hits the x-axis. We're looking for all the points. We want to know when that enzyme is not working. So when is the enzyme's graph, the enzyme's curve, not even visible? So in this case, it's anything below 4.75. It could be anything below 4.75. Like Janelle said, it could be 0, it could be 1, it could be 2, it could be 3, it could even be 4.5. That enzyme is not working. And then there's also another place on the graph, though, like Abigail and Sally told us, at 10.5, that enzyme is no longer working either. But also, it's not going to work at a pH of 11. It's not going to work at a pH of 12. It's not going to work at a pH of 13. So anything greater than 10.5 is where the enzyme will be denatured. So that's what we want to think about. It might be less than or greater than when it comes to denatured. And the optimum pH is going to be equal to something. We should be able to tell exactly what that optimum pH is. All right. Now, temperature is also another thing that we measured. So we've got a different graph here. Can someone tell me what the optimum temperature is for this enzyme? 40, good. 40 degrees Celsius. What about the denatured temperature? When is this enzyme going to be denatured? Let's think about our less than or greater than signs here. And thank you, JT, Melissa, Lisa, and Janelle for that answer. Okay, yeah, Josue, it's safe to say now, be careful, the pH actually can't be lower than 0. So we can't have a negative pH. But in this case, the enzyme is actually still working at a pH of 0. So this is an odd enzyme. Michelle, I'm saying pH, temperature in this case. We're not talking about pH. So we can have negative temperatures. That is true. We can have a negative temperature. Of course, that would mean typically that the solution, the water, is frozen. So we don't know, Josue, if it works below 0 or not, because we can't see in this graph. Michelle says greater than, Abigail says greater than 63. That is correct. That's what I said. I said 62. 62, 63. We can't tell exactly, but definitely greater than 63 degrees Celsius. This enzyme is going to be denatured. How do we know? We know because we can't see the curve anymore. The curve has hit the x-axis, and so anything greater than 63, anything to the right of 63 on this graph, not going to work. The enzyme is no longer working. It is denatured. Excellent. So we'll practice more with that. Let's see. So what's the optimal pH for pepsin here? Pepsin is a stomach enzyme that helps break down carbs in our food. What's the optimal pH for pepsin? 2.9, good. And what's the optimal pH for trypsin? Trypsin is found in the intestines. It helps break down lipids. So what's the optimal pH for trypsin? Okay, we've got, yeah, I guess we would say, yeah, I think this is basically, this graph is set up poorly, but 8.58, whatever, I see what you guys are saying. Correct, that is the optimal pH. Now we can see that they have basically no overlapping pHs. So pepsin is going to stop working at a pH of like 4.5 or 3.5, like 4 right here. It's not gonna work. Whereas trypsin does not even start to work until you get to a pH of about 5.5 or 6. So they don't have any overlapping pH here. All right, last question. The diagram here shows an enzyme substrate complex. This is something that I've asked you all to draw in your notes, similar to the diagram I asked you to draw in your notes. And it's also similar to questions that you all will see on your exam. So the diagram shows an enzyme substrate complex. Which of the following is represented by structure X? Is it the substrate? Is it the product? Is it the enzyme? Or is it the complex itself? So answers accepted in the chat. Is it A, B, C, or D? Thank you, Janelle. Anybody else? Thank you, Melissa. Thank you, Josue. Thank you, Abigail. We've got consensus here. Everybody is saying A. Thank you, Ashley and Lisa. A is correct. Yeah, the substrate. So how do we know? Well, we know because that's what we start off with. The substrate is your original reactant. It's what you start off with. Nothing happens to it in the beginning. But as it goes through the reaction, we see something happen to it. It gets digested. It gets broken down. So let me ask you another question. What is represented by structure Y here? And Ashley says it's the key as well. I like that analogy. C, yeah, good. Structure Y is the enzyme. How do you know that, Janelle or Melissa? How can you tell that structure Y is the enzyme? Okay, Ashley says it's the lot. I love that, Janelle. That's exactly what I was looking for. So structure Y stays the same throughout the entire process. We see it looks the exact same in the beginning and in the middle and in the end. That's what an enzyme, that's part of what makes an enzyme so special, is that it is reusable. Its shape is not changing. Structure X gets broken down into structure Z, so the shape is changing there. But the other thing we have in this diagram is structure Y. It's not changing. That means that it must be the enzyme. Excellent. All right, so we've got less than 15 minutes now. I didn't really assign anything new, but I did have someone sent me a question. I can't remember who it was, asking about an assignment that was previously called the honors performance task. I changed the name on Canvas. So now it's called U1D5 Biomolecules and Healthy Living Creative Project. I do want you all to do that. It shouldn't take too long. I'm not looking for, perfect works of art, but I like to give you guys, especially this class, opportunities to be creative because I loved your virtual lockers so much. So I want to keep giving you guys opportunities to kind of pull in different images and quotes and stuff that you're interested in and make things your own. So I think this assignment will give you an opportunity to be creative, and it'll be worth 40 points. So it's a pretty hefty assignment. I want you to do that. Other than that, I already had assigned the other assignment that I wanted you all to do, which is U1D5, Amoeba Sisters Enzyme Video and Quiz. So if you did that already, you're in a good place, as I know some of you already did. So go ahead and start working on this, as well as the exit ticket. Everything for this unit will be due on Friday, and we will have some work time on Friday for you guys to take some time to get caught up without us introducing anything new. But don't wait until then. Don't wait until Thursday night to start making progress. Use the time you've been given now, and that will, again, contribute to you all being organized and effective students. I'm going to put myself on mute. If you have any questions about anything, just let me know. Dijon, I was going to answer your question, but we are at 1:20 now, so everybody is good to go. I hope everyone has a good rest of your day. Stay safe, stay healthy, and I will talk to you all on Wednesday, tomorrow.
All right, see you tomorrow, Mr. Red.
Have a nice
See
day.
you. Peace out. Thank you. You too.